So thank you first to Governor and PAR for organizing this session. And both Governor and PAR asked me if I could give a small introduction about the field, about physics, and so I was happy to do so. And then later on, I'm going to talk about our field. And so since they wanted me to give an introduction, I felt it was to start understanding this between pharmacogenomics and pharmacogenetics. And so one of the things I did was go online into the dictionary and try to find the definitions for pharmacogenetics or pharmacogenomics. So pharmacogenetics is the study of genetic variation that gives rise to differing responses to drugs. Another definition that I saw later was studying an individual's genetic makeup in responses to a drug and guide prescription. However, it said also see pharmacogenomics, and so meaning that it's the same thing. So I went into pharmacogenomics, and it sounds like it's the same thing, the study of the effect of an individual genotype on the body and response to drugs. But then, going into this definition, seeing more of a difference, analyzing entire genomes, individuals, genetic factors, and so forth, but it also psychogenetics. And so finally, with this definition, I gave pharmacogenetics and pharmacogenetics tend to use interchangeably and exclusive. And basically, you can use either whatever you prefer. So in the history of pharmacogenomics and pharmacogenetics, this is a nice quote 62 from one of the fathers of pharmacogenetics, Werner Keller, who said that not all men react alike to a given dose of a given drug, nor do all animals. Variation of response from individual to individual of a given species has been regarded for a long time as a nuisance, as a threat, or merely a stumbling block on the road to the assessment of drug action. Now, this nuisance, if you think about it, is actually very, very important. And why is that? Well, it's thought that 12% of hospitalizations in the US are actually due to adverse drug effects, which is quite a lot. And if you are hospitalized in the US, you have a 7% chance of actually developing adverse drug reactions. In addition, it's estimated that pharmacogenomics, and adverse drug effects is the 5th leading cause of death in the modern world. And so that's quite a lot of importance. And 50% of adverse drug effects is actually thought to be genetic. So I think this is a very, very important health issue that not everybody realizes and how dangerous it is, and a lot of it is genetic. And in terms of the journey of a drug body, it goes through administration, absorption, distribution, target interaction, metabolism, and excretion. And in each one of these steps, you could have variation that influences how this drug is going through the body. And similar to mutations in genes, in terms of mutations that have been associated with pharmacogenomics, the majority of them have been coding sequences. Just because coding sequences are pretty easy to code. We know, for example, that CCC codes for choline. As changes to code, you change CCC to CT, change it to leucine. In our lab, not in mutants, regulatory genomic differences. And so here's one of the classic examples of this drug called Irinotecan that's used to treat colon cancer patients. When you give Irinotecan, it turns into this active form, SN38, that does the anti-tumor activity, and it is later, it gets cleared up in the liver by UGT1A1, which is an enzyme that turns it into this inactive form, and that gets cleared out in the bile. And so this enzyme, UGT1A1, has a promoter. And in 85% of Caucasian individuals, they have 6 repeats of a TA here in this promoter. 15% have an extra TA right here in the promoter. And those individuals, they have reduced expression of UGT1A1, and that leads to more of this active form, which leads to side effects, diarrhea, leukemia, and others. And now there's actually even a kit that you can purchase that can tell you if you have 6 repeats or 7. Based on that, you can decide about the dosage. And if you think about it 10 years from now, when we have genomes in our cell phones, we can scroll down to that position and see if we have 6 repeats or 7 repeats, and based on that, decide what dose to give to the patient. Now, it's important not only in terms of adverse drug effects, but in the future we can hopefully use the genomes to try and tailor drugs that will give much better therapy to the individual. So going back to this, the problem that we have is that like genes don't have a code. So even if it was a real element and I see a mutation in it, I have no idea of phenotype. In addition, we're really genes start with GT. Fine, and not is not encoded for protein, but we have important because if you and off that leads to phenotypes like malformations. So what can we do about it? Well, labs use genomics. We basically take chicken and fish for, in this case, 400. They might be functional to turn. We take a liver tissue. Proteins now bind to, and then we want to select antibody for this protein bind to all the sequences. You can try to figure out genome. What can high throughput curve? And we're looking for in the lab are mainly enhancer sequences. So just quickly, what an enhancer is, here we have this gene expressed in the limb, in a neural tube, and in the forebrain of a mouse embryo. And what people have seen is that if you inject just the promoter and the gene, you don't get this complex expression pattern. However, if you inject a bigger chunk of DNA, you start seeing this complex expression pattern. And the reason is that there are DNA sequences way out here called enhancers that turn the promoter and turn the gene on in the limb, another one in the neural tube, and another one in the brain. So combine these sequences and the promoter give you this complex expression pattern. So enhancers are sort of the promoter of the promoter. And so the way we functionally assay for them is by taking usually the human conserved sequence or a ChIP-seq-peak, sticking it in front of a minimal promoter and a reporter gene. And if this sequence is an enhancer, it turns on the minimal promoter, turns on the reporter gene. We make transgenic mice and harvest embryo at 11.5, stem for Z, and in this case I'm showing you a very consider. The problem with mice is that they're very B, they're not good, and C, you're only looking at 1.5, and you have to take the embryos every time to look differently and also later on. And so in my lab, we set up also doing exactly the same thing, but a zebrafish, and inject, and we do this in a high throughput manner over time, and here I'm going to do a fin enhancer similar. Now for the project that I'll talk about in a minute, we're actually very interested in different enhancers, and so there's a third technique called hydrodynamic tail vein injection. In this technique, you can inject a large volume of liquid, about 10% of a mouse weight, so about, if a mouse weighs about 30 grams, 3 ml. This is relevant to me. If you inject this to me, it will be about 6 liters of liquid in a very short period of 4 to 8 seconds. And so the heart can't handle it, and you get very, very specific expression in the liver. And here I'm showing you a nude mouse that was injected with GFP. You can see that this is very, very specific to the liver. So using this technique, we can assay, for example, for enhancers in the liver. If the sequence liver enhancer turns on the minimal promoter, turns on the reporter gene, it will light up in the liver. So we're interested in, and this is in collaboration with Kathy Giacomini, the others, are the liver membrane transporters that are really into the liver, and outside the liver. One is one of the transporters for metformin, which is the number 1 drug in the market. And you have mutations in OCT1. As you can see here, you get less intake into the liver of metformin, and that doesn't really help the patient if you give them metformin. On the opposite side, you can have mutations that lead to more expression of metformin, more intake of metformin, and that could lead to side effects. So go ahead, you can think about similar things for regulatory elements. If you have a regulatory element that tells OCT1 to be on the liver here, if you have mutations that you can much metformin go into the liver, and that leads to side effects, to less of OCT1, which will lead to for the patient metformin. To assay for enhancers of the different liver membrane transporter technique that I talked about previously, and I just want to show that you can use this technique for other things, not just enhancer assays. So for example, you could use it for coding sequences, giving one mouse the gene, the cDNA, and another mouse the gene with one variant that you think affects drug intake. Then you can give a labeled drug and see if there's any difference in intake between this one and this one. You can use it for promoter sequences where you give a promoter and a promoter in the variant to another mouse and see if there's any difference, or you can use it for enhancer assays. And this is just one example of how we use it for promoter assays, and also I'll later get into enhancer sequences, which are more interesting, the most interesting to us. So in terms of promoter assays, here's a membrane transporter, SLC28A2, nucleoside transport into the liver. And using in vitro assays in HepG2 cell lines, there was this haplotype here that gave much more increased expression of this reporter gene, the promoter of this membrane transporters. And so we went and used exactly the same haplotypes in vivo using our technique. And as you can see, we got very similar results, showing that we can use this technique and obtain very similar results using a promoter assay. In terms of enhancer sequences, what we set out to do was we took 9 membrane transporters, these interactions with these important drugs. And we did what comparative using by Methotrexate, where we go through peaks, and what we're trying to find is peaks, and this human, monkey, and human and mouse, we're trying to find peaks that are pink here, but not which are coding sequences. There are conserved non-coding sequences. And we looked at all 9 different genes. We found about 622 conserved non-coding sequences, or ECRs. And so that's what I tested in mouse enhancer assay. And so the next step was using transcription factor binding sites as a way to rank check first. So we look for very specific transcription factor binding 22 sequences, and we rank them based on the balance of these transcription factor binding sites. We then took the vector that has small promoter and reported what we see there's any one of these 50 sequences is an enhancer compared to the negative control, an empty vector. So we inject these into mice, and then point later check to see if we get expression from this sequence compared to the negative control. And as a result, from all 50 sequences, we initially injected, and this is our negative control PG, the empty vector. No liver enhancer, ApoE, that you can see gets. And you can see we got a few other sequences, about 12 out of source significance. And since this assay is very variable, up to at least in mice, to check to see if it's consistent. About 5 out of the 12 sequences that were initially pretty strong were still significant when we raised the number of mice. Now, since this is a pharmacogenomics assay, the next step is to see if there's any variation and then see if there's any difference. And so what's nice about this collaborative project is that cohort, 1,000 people, healthy human volunteers in the San Francisco Bay Area that are racially and ethnically diverse from different populations, African American, Caucasian. And what's great about them also is that they can be into the lab and for genotype, phenotype drug studies. And so what we did was sequence these for the enhancers that we found. An example of one of these for ECR35, how that went through. So ECR35 lies here in an intron between these two membrane transporters, liver membrane transporters. Both of them interact with a lot of the drugs, as you see here. And when we sequence different haplotypes within it. So the next is a difference with haplotype and comparing it to a different sequence. And so we did these injections. And here, this is our negative control. This is our ApoE, positive liver enhancer. This is our reference sequence. And you can see that these two haplotypes gave significantly reduced expression of our reporter gene. It's worth mentioning that these haplotypes, some have a high minor allele frequency. You can see here in Caucasians, African American, Asian, and Mexicans, that this haplotype star 3 is actually pretty high in some of these populations. And so the following is we have back to 88 Caucasian human liver tissues. And so what we can do with those is genotype them for this enhancer. And in addition to real-time PCR on these two genes to see if there's a correlation between the variants that we find and the expression levels of these genes. And so when we did that, we saw that there was no correlation with this gene, but we did observe a correlation between the variants in this enhancer and the expression levels of SLC01A2. And so just to remind you, these are the results that we got from our enhancer assay. And so for this star 1, star 3, we didn't see a significant difference. And that was similar in the expression pattern for SLC01A2, where we don't see any significant difference in terms of expression. For star 3, this was a very low haplotype, and so we only had one individual from the 88 that actually had that change, so we couldn't do this assay. But for star 3, we had sufficient individuals, and as you can see, we see reduced expression of SLC01A2 with this SNP, and similar to the results that we got for the enhancer assay, and suggesting that this SNP here may lower the expression levels of this gene. Interestingly, in this region, there was a GWAS study that found associations, clearance, and this region over right here. And previous work has shown that have coding mutations in SLC01A2, the gene where our enhancer reduced methotrexate clearance due to these. So both of these are actually pretty promising in terms of possibly to be involved in methotrexate clearance. And so we've contacted Maryland, who did this GWAS study, and we're now typing for patients to see if there really is a correlation between on here and methotrexate clearance in that region. Future actions are sequencing additional liver enhancers and carrying out functional analysis on these variants. We're doing association studies of gene expression, as I showed you, with liver enhancer variants in human liver tissue. We're wanting to do clinical genotype-phenotype drug studies, and one example is the thing that we're doing with Methotrexate currently, and we're starting to use seq approaches to identify additional enhancers that are associated with pharmacogenomics. And I thank members of my lab and E.J. Kim, a graduate student who's trying to do a lot of seq on this, and all the people in part of this collaborative projects, Steve Brenner and Kathleen Mediana Kratz. Thank you very much.
Now we have time.
Nice talk. So, do the systems that you did these mouse developing mice and then fish to simply?
And no, so for this tail nest, because we're really interested in the liver, we have fish and mouse. There are other liver enhancers that we've tested in zebrafish in the liver, and now we're testing. It's not the project, but
do you have any more questions? And all presentation, and we're waiting the setup for